Hi everyone. I would like to present polymersomes as stable nanocarriers for a highly immunogenic and durable SARS-CoV-2 protein subunit vaccine. To introduce the nano delivery systems, there are three classifications: polymeric, inorganic, and lipid-based nanoparticles or lipid-based nano delivery systems. With lipids, it's easier and you have a range of physical chemical characteristics, highly bioavailable because it's all lipid based, and payload is flexible. The encapsulation can be tricky depending on the molecules that you have selected. Whereas inorganic particles, it's widely used in theranostics or in vitro assays. The properties can be tuned based on the surface and structure, but in vivo toxicity and solubility is not well characterized. When it comes to polymeric base, there are polymersomes, dendrimers, polymeric micelles, and nanosphere. Since it's a polymer based, it offers high flexibility in terms of loading and the design of molecules, and for example, easy surface modification. But there is potential concern of possibility for aggregation and toxicity. In the further slides, I will be showing mostly about the polymersome and its comparison with liposomes. What we work at ACM Biolabs is Polymersone Platform. Essentially, they are spherical vesicles. So you have an hydrophobic core made of amphiphilic block of polymers, and you can incorporate hydrophobic molecules in the membrane. At the same time, you can encapsulate hydrophilic compounds, including biological macromolecules such as proteins and peptides in the core. These are self-assembled from a hydrophobic and hydrophilic block of polymer into spherical vesicles. Depending on the block lengths, you can form vesicles, micelles, and rod-like structures. One of the biggest advantages is these hydrophobic block and hydrophilic block can be tuned based on the chemistry that you use. And as long as you are within the ratio of between the hydrophobic and hydrophilic block, you can form vesicles, but of different membrane thickness. So I have shown here two examples of vesicular structures, how the membrane properties can be tweaked or the thickness can be maintained. When it compares to lipids, as we all know, it's a single size, 3 to 5 nanometers. And you have, that's why lipids are regulated in most cases to avoid any of lipoprotein attacks. Whereas polymers, it is already the PEG is the hydrophilic part and the hydrophobic one that you can actually adjust, increase or decrease based on the requirements. So essentially, polymersomes offer a minimalist, modular, and versatile, and you have the ability to tune the block of polymers. Now, to compare both polymersomes and liposomes in terms of physical chemistry properties, liposomes, as is given, it's a bilayer architecture made of lipids, whereas polymersomes, it's also a bilayer architecture made of polymers. In terms of encapsulation, both can encapsulate. And for lipids, you have earth permeability because of the lateral mobility of these lipid membranes. Whereas here you have low permeability because the molecules you have much lower lateral fluidity as opposed to liposomes. The combination, when you look at the lipids, that it has low stability, whereas in polymers that you have high stability. For applications, you can take polymersome as a template or polymer as a template, and then you kind of get best of both worlds in order to make available for encapsulation and yet low permeability, low lateral fluidity, but at the end, high stability. Now, with these toolbox, what we do essentially, we take the polymers and lipids and we can we can form vesicular structures. As mentioned, we can actually integrate hydrophobic molecules in the interface or in the lumen hydrophilic molecules, or you can do the surface functionalization. So for the past 15 years, we have developed an array of different chemistries that we can use for actually integrating proteins, peptides, and oligos in the polymersomes as delivery vehicles. To give you some examples, we can insert by lipids, or membrane proteins can be inserted, or you can conjugate by a simple functionalization, or you can actually add hydrophobic molecules like what we are showing here to the polymersome. Without changing the physical property of these polymersome, you can load different type of molecules in it bilayer onto the surface. Similarly, you can encapsulate proteins or peptides or small molecules or DNA and mRNA in the lumen. Again, the surface charge or the properties of the polymersomes is not changed without changing the properties of the polymersomes. Now, moving on to how we are using polymersomes as antigen delivery. This is schematic representation of an hairy tennis ball, but essentially what you have is an amphiphilic block of polymer self-assembly. When you do the cross-section of it, you will have a bilayer architecture, and you can encapsulate the proteins or peptides, or you can actually incorporate the proteins or the peptides on the surface. So when we started this basic science, or when we started translating this basic science as a technology platform, we were offering this platform to companies that we could do a potential application as vaccine delivery, or to generate monoclonal antibodies, that you are able to deliver an antigen stably. So during those initial days of discussion, we have been introduced to PEDV. As many of you know, post-epidemic diarrhea virus is a coronavirus. It's alpha coronavirus, first isolated in 1971. Now it's spread worldwide. You have a spike protein, and you have envelope protein in the membrane, and the bilayer architecture, the nuclear protein, and the genomic RNA. So these PEDV, if you have best practices, you can limit the outbreaks, but PEDV is still an issue in some Asian countries. The PEDV attacks the young piglets of day 3 and day 10 old. And once you have PEDV, the mortality is 80 to 100%. So when we were discussing about the platform, one of the questions that we have been asked is, can you actually make these PEDV using a nanoparticle as vaccine? So we have taken the spike protein, and we have incorporated into a polymersome, and we have given to our partner. So they have tested this as to just let you know, PEDV is a gastrointestinal coronavirus. So this causes gastrointestinal disease. So you have huge diarrhea in the piglets, and the piglets will have diarrhea, and then they will die. So what we have done is we did prime on day 1 and on day 14, and challenge was established on day 28, and we look at the clinical score. Essentially, you have a diarrhea, or you have a normal stool. So the vaccine was given 2 different routes, orally and intradermal, and it was given in weaning pigs of 7 to 8 kilograms. And what we have seen, this was done in a place where you don't have the luxury to measure serology. But what we were looking at is actually the clinical score. 1 means good, 4 means bad, that you have a heavy diarrhea. And as a control, ACM alone, without no antigen, it works as placebo. So you have high diarrhea. When you have ACM antigen, it does, but when you formulate with adjuvant, you still see a titer or a normal feces or fecal score. So similarly, when we did intradermal, again, the ACM control alone shows high diarrheal content, and ACM antigen better. When it comes to ACM with adjuvant, you have a better response. So based on this, our vets told it is better to go for PEDV oral administration because you need to pass on the maternal immunity to the piglets. So based on this, we decided to go to the sows. So here, the sows were given vaccine at the age just before farrowing. And during that, there was a natural infection in the farm. So there were pigs or sows of different in pans, and they were having piglets born at different time points. So what we have seen, or what our partners have observed, is that the vaccinated mothers, or the piglets born to vaccinated mothers, you have high protection, whereas the piglets born to non-vaccinated, you have low protection and hence high mortality. As the age goes, the difference becomes not so substantial. And at the age of 14, you have much low mortality rate. So this was exciting, but this was done in a low academic settings, in academic settings. Now we have to demonstrate this in a regulatory settings, and currently the vaccine and we are planning to go for the USDA registration. That means the entire process of manufacturing has to be established in the US, and also the challenge models and the challenge studies have to be established with our current vaccine. Now, this kind of gave us an idea on the platform, the capability of the platform. So we were wondering, how come a 140 kilogram of animal injected with a tiny amount of polymersomes is doing something? So then we wanted to understand what's happening when you inject polymersomes. Then we look at, obviously, we didn't do mechanism for study experiments with pigs. Then we took a polymersome labeled with a dye, rhodamine, and we administered into mice subcutaneous. So what we have seen, out of all the cells, polymersomes are taken up efficiently by the CDC1 and CDC2 of the dendritic cells. So we have looked into this with multiple administration routes, IM, subcutaneous, and intraperitoneal. In all cases, the polymersomes are labeled with rhodamine. These are all polymersomes are labeled with rhodamine, are efficiently taken up by the DC1 and DC2. So this kind of intriguing. We looked at, okay, this is empty polymersomes, and the polymersomes are labeled on the surface. Now, what happens when you include a cargo, and whether it is taken up by the same DCs, and also to see that the polymersomes are acting as immunogen other than just an efficient delivery system. So for these experiments, we have taken 3 different samples: polymersomes alone, CpG as an immunostimulant or an adjuvant, or polymersomes encapsulating CpG. So what we have seen, the polymersomes alone, no upregulation on activation markers on CDC from draining lymph nodes. So to prove that, the polymersomes alone, we don't see any activation. Thus, it is non-immunogenic. Free CpG, some activation on CDC2, but not on CDC1. So you see some activation on the CDC2, but not on CDC1. The difference here. But once you have in polymersomes, you can see both activation in CDC2 and in CDC1. This actually suggests that you have an efficient delivery of CpG to the endosomal TLR9. So, based on our experience in PEDV and also to understand the mechanism of action. When the COVID hit, like multiple research organizations, we looked at the spike protein and say, okay, this is a BSL-3 vaccine, so you need to have a different platform to produce the vaccine. So for COVID vaccine, we took the spike protein. Obviously, we were working with PEDV, which is alpha coronavirus. This spike protein has been well established, well characterized, and we encapsulate spike protein in a polymersome, and then we encapsulated CpG as an adjuvant in polymersome. This is just, I'm not going to show all the data, but we have demonstrated that, mixing these vesicles will give exactly the same immune response rather than having them together. So our vaccine, for ease of manufacturing large scale, we produce the ACM antigen and ACM CpG, and it will be mixed, and the dose is 0.5 ml in a pre-filled syringe. That's our vaccine design. So we looked at different constructs. So we have taken S1, S2 without the TM domain, and then we have taken the trimer with the trimerization domain. S1, S2 is without the TM domain and S2. We established ACE2 binding assay, and we looked at whether, first of all, we saw the expression of the protein, and this is our in-house purified S1, S2 in Cypro and in Western blot. And these proteins are produced using insect cell expression system. And then you compare with the S2 and also the commercial trimer that we have produced, this particular one, with the primerization domain. So we looked at the ACE2 binding, and what we wanted to measure is, after encapsulation, do you see any difference in the ACE2 binding? So what we can say is, before on day 1 and week 20, you have similar EC50 levels, but after encapsulation, they are lysed to actually present. Sorry about that. So they were lysed and made sure that you have exposed the protein, and you measure the functionality of the protein to ACE2. So this actually confirms that the self-assembly process or the fabrication of protein in polymersome doesn't change its ACE2 binding ability. So we have taken these vaccines and we have immunized in mice, subcutaneous prime boost. We looked at IgG titer, total IgG, neutralizing response. So we are using a CPAS competitive ELISA, which is a product developed by Duke NUS in Singapore, as opposed to live virus neutralization. So this can be used as a surrogate study for live virus neutralization. And what we did is immunize on day 0 and boost on 14, and the bleeds were taken at these time points. We compared free protein or the spike 2 versus ACM encapsulated one, and we looked at free trimer versus ACM trimer, and we looked at free spike protein S1, S2 versus ACM S1, S2, and the final vaccine, which is the free protein, free CpG versus the ACM encapsulated protein and ACM CpG. What you can see here, in all cases in bleed 2 or bleed 1, there is a difference between with and without encapsulating the protein. And once you are administering with CpG, you see much tighter immune response, and you have we have done one limited study of 0.5 microgram, let's say much lower dose, and we still see similar responses. So this to prove that ACM polymersome delivered spike protein is enhancing the immunogenicity. So this is on the total IgG. Then we looked at whether is it the same when it comes to the neutralizing titers. And what we have seen here, we looked at, as I said, this CPAS kit. Again, the same different set of animals, free versus S2 protein encapsulated, and trimer, free versus ACM trimer, and the free versus ACM S1S2, and the final vaccine. What we have seen is we have a sustained neutralizing activity for this particular vaccine at 5 microgram of antigen and 5 microgram of CpG that was administered in ACM. There were declining responses for other groups, and this was prominent to show that after 40 days after boost. In parallel, we did live virus neutralization, and here what we have seen is essentially that the ACM group is the best performing group comparing to all these other samples that we have measured. So this to us prove that ACM COVID vaccine is producing strong, durable neutralizing titers. Then we looked into the T-cell responses, and what we have seen is that the spinocytes were harvested and stimulated with spike protein multi-epitope peptides, and we looked at functional CD4 and CD8 T cells 40 days after boost, and we see that IFN gamma for both the vaccine samples in both CD44 and CD4 T cell memory T cells, CD4 T cells, and memory CD8 T cell responses. And you don't have a Th2 response. And again, when you look at IL-5, for example, the free protein is giving an IL-5 signal, but once you have encapsulated in ACM, ACM S1S2 and ACM, it goes down. That's also shown in the free protein with CpG. So based on all of this data, our COVID vaccine that we have with CpG and a spike protein as a potential vaccine for further development. So with this, I would like to conclude. So I hope I have demonstrated that ACM vaccine is eliciting robust neutralizing titers that persist at least 40 days. And we have also performed GLP tox studies in New Zealand white rabbits, and we have observed no adverse effect level with ACM COVID vaccine. We are aiming to enter clinics by 2021, Q4, with the beta variant as a booster vaccine. Plus the platform, I have shown the examples of the flexibility of the platform, that it can be used as a plug and play, not only for proteins, but also peptides and mRNA. With this, I would like to thank you for your time and happy to take any questions. Thank you.